So, hello everyone. Am I audible? Can you hear me?
Yes, sir.
Yes, sir.
Okay,
Good
so
evening, sir.
yeah, so good evening everyone. So, everyone joined or someone else left to join? Any idea? Okay, so I think everyone joined because it's already 6 7. So, we can first of all welcome to the first uh, live session with you regarding the course NOC 24 BT 63 I think immunology course. So I think the week one lecture you have finished, right? May I get a confirmation from you that all of you watched the week one lecture?
Yes, sir.
Yes, sir.
Okay, nice. So so in this session, basically the purpose of those sessions are to revisit the contents you have uh, went throughout the lecture in the of every week, and if you have any doubt you can ask your doubts and you can clarify your doubts. So please feel free to clarify your doubts regarding the weekly topics, and we will discuss some question and answer so that it will help you to solve the assignment of this of the current week, right? So how many lectures did you attend? Five, right?
Yes, sir.
Okay, so from week one there was five lectures. So we can start now. So I will share my PPT and you will let me know whether you can see it or not. Then we can start our discussion. Okay. So in this week, basically, so can you see the PPT slide? You can see.
Yes, sir.
Yes, sir.
Okay, fine. Okay, so let's start. I think someone is. Okay, so the basically this weekly topic was the topics for week one was about the immune cell, a basic introduction about immunology, right? So I will just so I would like to start with a question that what do you understand with immunology, and what is your if I will ask you to define immunology in a one sentence, then how will you do? Anyone will come?
The study of
Study of
the
yes,
study of immune system.
study of immune study of immune system. So what do you mean by immune system?
The
And
immune system protects our body from the pathogens and foreign invaders. Because of these invaders, we can able to obtain many diseases. So this immune system can prevent the diseases. That is the function of our immune system.
okay, fine. So if I will ask you now one thing, if I just modify your, I mean, my question that, so you are saying that to protect ourselves from the foreign particle, right? So now if suppose you are inhaling a pathogen, right? Right now, so yeah. So after that, first you were suppose first you were infected with a specific strain of a pathogen, for example, COVID, right? So what is the possibility or what will be the outcome that when the second time when the same pathogen, exactly the same strain will infect you, so will you fall ill, or what will your body do then if the same strain will again infect you? Any idea? Hello? Anyone?
Hello?
Yes.
So secondary immune response, so it will form the antibodies. So it is for the lifelong.
Sorry? It will produce antibody. What? Okay. Arpit is writing faster immune response due to memory cell. Very good. So basically, because you were previously infected with the same foreign particle or pathogen, that's why you already have the antibody corresponding to that particular pathogen. So of course your immune system will be activated, they will start uh, doing their work, but the thing is you will but now this time it will be a secondary immune response, right? For secondary immune response you will get a more rapid, more intense immune and more strong immune reaction. So that is immunology. When we study the immune system, and the need of studying this immune system is because this system is required to protect us from foreign particles or pathogens, right? So this foreign particle or pathogen which you were saying, so should it be always a living thing, or it can be non-living thing also? Will your immune system be activated only you were infected with a living thing, or I mean only you were infected with some bacteria or virus or fungi, or it can be induced if you were inhaling something else kind of non-living? Is it possible? Is it possible?
Yes, we can able to protect our body from the pathogens.
No, but I'm not asking for pathogen. Suppose, for example, you are walking in a road, road full of dust. Then suddenly a wind come and a lot of dust will come to your eyes. So then, what will your body do first? Da-
The eye will stop the security tears
I- eyes will start.
to security tears, and that can able to remove the dust from our eyes.
Yes. So first of all, the body will do is it will just blink your eyes, right? This is the first defense, so that this dust particle cannot enter into your eyes. Now, if some of the dust particles will manage to enter into our eyes, then what will happen? The eye will start secreting the lacrimal fluid, right, which has acidic pH. So that will basically remove the uh, dust or whatever it is from your eyes. So so this is also example of your immune system, which is called as innate immune system, which is responsible to protect you from dust or non-living things, right? So that is the integrity of the immune system. It can protect you from any foreign particle, right? So now if we start, then uh, as this week content covered only about the introduction of the immune system. So we can first start with immune cells. So how many immune cells you are known about? Anyone?
Four types of immune cells are there.
Which are they?
B cells, T cells, NK natural killers,
Huh.
I and C cells.
B cell, T cell, natural killer cell, and
So macrophage, dendritic cells,
yes, macrophage.
neutrophils, these are the cells of innate immunity.
Yes. Acha. So why
Mast cell.
mast cell? Yes, very good. Basophil mast cell, right? So why are you saying
Basophil cells,
yes, 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 yes? Fine. So just someone told me that this is innate immune system. Why it is innate? And what's the difference between innate and adaptive immune system? Why it is innate? This macrophage mediated, dendritic cell mediated immune system. Why is it innate? Why do you call it innate? Any reason? Hello? Okay. Yeah. So anyone want to come with a guess?
innate immunity is our body to general protection mechanism. When innate immunity fails, if the infection still exists in our body, to another system which takes some time but make a definite and specific weapon to protect the infection.
Yeah, but how do you define innate immune system? Like, for example, someone told that this neutrophil, macrophage, these are innate immune system. So why are they innate? Okay, can someone please mute the mic if the background is okay. There is a noise from the background. Can that person please mute the mic? Thank you. Okay, so now the question is, how do you define the innate immune system? So someone told me that yeah. Tell me.
Sir, innate immunity is non-specific.
Non-specific, very good.
Basically, it's provide the general protection.
General protection, yes. Anything else except the non-specific?
It activates before the antigen expulsion.
Very good.
And
So
adaptive
yes. So true.
innate immunity means
Ha.
that is obtained from that is already in our body by birth. That is known as innate immunity. Innate means
Yes.
born.
Yes. Innate means which is already there. So two things are very important that to define innate immunity. One is it is non-specific, right? And second is it is already there. It does not need any kind of training, right? It is there inside your body. It knows how to react, how to act, to protect yourself. It already knows. So that's why it is innate immune system. Why? Because, so let's come to the point, non-specific. Why is it non-specific? It will not differentiate between the antigens, between the, the pathogens which will come, right? It will just non-specifically catch and kill, and at least try to kill that particular organisms. It will not differentiate. That's why it is non-specific, right? Second thing is, it is already there, so you don't need to train it. Like for example, adaptive immunity system, the name itself suggests that it has to adapt, right? It has to adapt, it has to acquire some training so that it can combat with the pathogen. That's why it is adaptive immune system. The very good example is antibody, right? So that requires the training, the requirement of the training here. But innate immune system does not need any requirement of training, right? So that's why it is innate immune system. Someone, I think, in the queue. Let me just, yeah. So that is innate immune system. So then, what is adaptive immune system and how does it work? Very briefly. Adaptive immune system, and how is it different from innate immune system?
Adaptive immune system activates on when the body is exposed to a external
Yes. Antigens.
like antigen. Yeah.
Okay. So and after
So
that,
body will produce the antibody.
Huh.
Yeah. Body will produce the antibody and will be getting a immunity against that particular antigen.
yeah. So why the name is adaptive here?
Because it is not present in the body as
Yes.
such. When we are
Yes.
exposing to a external condition, that immunity.
Yes. So it requires a training, right? So later we will discuss how this training happened and how your immune system knows which antibody to produce and when to produce. Okay. So now in this slide we can focus on the immune cells. There are all immune cells, or any cells, they start originating from the stem cell. Okay. So what is stem cell?
Stem.
The cell where all cells are generated.
Stem cells are?
It is the precursor cell where the all cells are generated.
Yeah. So it will basically, it has the capability to self renewal activity, and it can differentiate to different cells, right? This is the that's why it is sometimes it is called as a progenitor cell. So the immune cells are originated from a stem cell. Which kind of stem cell? Hematopoietic stem cell, right? Hematopoietic, it comes from that this stem cells reside in the blood. That's why. So this hematopoietic stem cell will first differentiate into two progenitor cell. Huh? So this hematopoietic stem cell will first differentiate into two progenitor cell. One is myeloid progenitor, another thing is lymphoid progenitor, right? So now myeloid progenitor, again it will differentiate into different cell. One, for example, it will be differentiated into the dendritic cell, right? Apart from that, it can be differentiated into granulocyte, for example, monocyte, eosinophil, basophil, megakaryocyte, and erythroid progenitor, right? So this myeloid progenitor, it can differentiate it into dendritic cell, as well as it can differentiate it into the progenitor of monocyte cell, eosinophil, basophil, erythroid, right? And from these progenitor cells, these cells will be differentiated into, for example, this monocyte, it will be differentiated into neutrophil, eosinophil will be differentiated into the eosinophil. Basophil progenitor will be differentiated into basophil, and this mega karyocyte, this is a progenitor cell, it will be differentiated into the platelets, and this erythroid progenitor cell, it will be differentiated into the erythrocytes. Clear? So this is the part of the myeloid progenitor. Another.
Can you zoom it? Can you zoom it a little bit?
Yeah, sure. Is it fine? Is it fine? Okay. So hematopoietic stem cell, as I discussed with you, it will first be differentiated into the myeloid progenitor, and then this myeloid progenitor will give rise to dendritic cell and this monocyte, neutrophil, eosinophil, basophil, platelet, and erythrocyte. Another population it will give birth is lymphoid progenitor, right? Why this lymphoid name comes? What do you mean by this lymphoid? Anyone?
Lymphoid tissues or circulates in the lymphatic vessels,
Yes, lymphatic
tissues, and the that is protecting our body.
Yes, it is. Yeah, so they are basically present in the lymphatic tissue and lymph node, right? So they are lymph based. That's why they are called lymphoid progenitor, right? So this lymphoid progenitor will differentiate into the natural killer cell or NK cell, and this lymphoid progenitor can be differentiated into the two progenitor cell, that is B cell progenitor and T cell progenitor. T cell progenitor will be differentiated into TH cell or T helper cell and cytotoxic T cell or TC cell, and B cell progenitor will be differentiated into the activated B cell or actually B cell, right? Apart from that, this lymphoid progenitor will be differentiated into dendritic cell also. So again, myeloid cell will give you dendritic cell, all of this granulocyte, right? This lymphoid progenitor will give you NK cell, helper T cell, cytotoxic T cell, B cell, and dendritic cell. All of them are lymphatic cells, right? They are the part of the lymphatic system. Okay. So now if I will now if I will ask you that, or if the question comes with the exam, that dendritic cell is coming from which progenitor cell?
Both the lineages.
Yes, both
Myeloid
the
and
lineages. Yes, one is from myeloid, one is for lymphoid. So that's why it is very important, because it is common. It is common for that it will be generated from myeloid as well as the lymphoid progenitor cell. And natural killer cells comes from which progenitor cell?
Lymphoid progenitor.
Lymphoid. Yes. And can you name a anuclear cell and its progenitor? Can you name one anuclear cell? Anuclear means when there is no nucleus. So can you name one anuclear cell and its progenitor? Anyone?
Monocytes.
Sorry? Monocyte is not.
Monocytes. Erythrocytes.
Yes, erythrocyte is the nucleus-less cell, and its progenitor from which lineage? From which lineage?
Myeloid.
Myeloid. Very good. So, what do you expect, that this erythrocyte, platelet, basophil, eosinophil, neutrophil, all of them are from myeloid progenitor. So they should stay where? Lymph system or blood system?
system.
Yes, blood system. Right? So, yeah, so any doubt here? From this slide, any doubt?
Sir, I have doubts, sir.
Yes, yes, tell me.
Sir, if the sir, if RBC are
RBC?
progenitor, am I right?
Sorry?
Where the RBCs are formed, sir?
I didn't hear your question. Can you repeat, please?
So where the RBCs are formed?
Erythrocyte is RBC, na?
All the blood cells, all the red blood cells.
Yeah. So erythrocytes are RBC, right? So it,
Okay.
yeah. So it is coming from myeloid. Myeloid progenitor. From myeloid to erythroid progenitor. From this, it will come to erythrocyte. Clear?
Okay. Another granulocyte, monocyte, eosinophil, basophil,
Yeah. Yeah.
both are from the myeloid progenitor.
Yeah.
Right?
Yeah. All of them have the myeloid progenitor. Yes, you are right.
Okay. Platelets are is the platelets are present in the erythrocyte, I mean RBCs.
Platelet are also coming from myeloid.
Okay. Where the platelets are present now?
Okay, all of these things are present in the blood. Erythrocyte, platelet, and these are of course, you know, what should I say? WBC. This neutrophil, neutrophil is what kind of WBC, eosinophil, basophil, all of them are WBC, but we specifically we call them neutrophil, eosinophil, basophil. So all of them are part of blood,
Okay.
right? And this monocyte, when it goes into tissue, then it will be more differentiated into macrophage. I think you have heard about. I think you have heard about the macrophage. So this monocyte, when it goes into the tissue, in the blood, it is known as monocyte. When it resides in the blood, this monocyte, then it acts as a monocyte. But when it enters into the tissue, then it will become macrophage. There is more such differentiation, and it will become macrophage, which is a phagocytic cell, right? It can, if it has any, if it find any kind of, you know, foreign particle vicinity to them, then they can just phagocytosis. What is phagocytosis? In one word, yeah, or in one sentence?
Engulfment of the foreign particle.
Yes, very good. So if if it will found any, you know, foreign particle in the vicinity of it, it will do the phagocytosis. A macrophage is famous for that. Okay. So any more doubt from this slide? Should I go ahead?
Yes, sir.
Okay. So next we will start with innate immunity, as I think you have already known about that. So the member of the innate immunity is skin. First of all, it is a mechanical barrier, and the very good example is when you are exposed to a very high intensity light, then your eyelid, then your eye will automatically closed, right? Your eyelids will come down, it will blink. That is a very good example of the mechanical barrier, which is your eyelid. And also the skin. The skin, it acts as a mechanical barrier which prevents the entry of the microbes. Why? Why does so? Skin, why? How can skin act as a mechanical barrier? Suppose any infection, any, you know, pathogen comes to your skin, then what will happen in your skin? If a mosquito bites you, then what is the changes you saw in your skin? Or if any sits on your skin, then what is the changes you see in your skin? Anyone, please?
Sir, inflammation or swelling.
Inflammation. Yes, very good. So why this inflammation happens? But the thing is, I'm asking about skin. Okay, but this inflammation is happening in your blood system, right? So we will discuss that. But in your skin, how does skin protect you from the bacterial or viral or fungi infection? When you sweat, right? All of us sweat. So during this sweating, what is the pH of the sweat? Acidic, basic, neutral?
Sir, acidic.
Acidic. Yeah. So that's why this.
3.5 to 4.5.
Very good. So that's why this acidic pH prevents the growth of the microbes. Whenever microbe will come, they will expose to acidic pH because you have sweat in your skin, and this sweat secretion will retards the growth of the microbes. That is mucosal membranes. So this is actually known for mucus. It is a very important part of innate immune system. It can entrap the foreign particle, right? It will entrap the foreign particle. For example, when you cough, why do you cough? Because in your respiratory tract, when it will be infected by some virus or pathogen, then your respiratory tract will start secreting mucus, right, inside your tract. So after secreting of mucus, what will happen? This mucus, it will entrap the pathogen, so that pathogen will be immobile. It will be immobilized, so it cannot move after that. When it will be entrapped by the by the mucus, right? So now that's why it prevents the spreading of the infection, because it will entrap the pathogen, and then this entrapment will cause you coughing. So then when you cough, then your pathogen along with the mucus will come
Sir,
out.
your voice is not audible.
Hello? Am I not audible? Am I audible? Hello?
So, is it okay? Little bit clumsy. Your voice is audible. Little bit clumsy. Is it audible?
Okay, okay. Right now? Is it clear?
Clear, sir.
Okay, okay. Thank you for the confirmation. So yeah, so I was talking about the role of the mucus. Mucus, what does it do? It will entrapment the pathogen. Suppose your respiratory tract is infected with a pathogen or virus, then this virus will be entrapped by the mucus inside your tract, so that this virus cannot further spread, right? So then when your mucus will entrap the virus, then you will cough, you will start coughing. So because of this coughing, your mucus along with the entrapped virus or bacteria, it will come out from your throat, right? So that's why it gives you the protection against the respiratory tract infection. These are anatomic barrier. Physiological barrier: temperature. Normal body temperature which we have, that already inhibits growth of some pathogens, some of the pathogens. And fever: when you were infected by virus or bacteria, then you will start having fever. This fever will respond. This fever means your temperature will be increased. So this high temperature will inhibit growth of some pathogens. Low pH: when your food, suppose the food you were taking, that food is contaminated, it is infected by some pathogen, right? So if it happens, and of course the food we have, so it is not possible that this food will be although it is cooked, but still there might be a trace of some pathogens. So if some pathogens will go along with our food inside our stomach, then the acidic pH of the stomach that will kills most of the ingested microbes. Then chemical mediator, one is lysozymes. We because we have the lysozyme enzyme which cleaves the bacterial cell wall. Interferon, it will induces antiviral response. Complement lysis, it will facilitate the phagocytosis. TLR are toll-like receptor, they will recognize the microbial molecule and it will induce the secretion of the immunostimulatory cytokines. Then phagocytic barrier, various cells internalize or endocytosis and break down the foreign macromolecules. The example is macrophage. Okay, and some specialized cells, like for example blood monocyte, neutrophil, they internalize, kill and digest whole microbes, right? And the example is macrophage. Inflammatory barriers, tissue damage and tissue damage and infection induce leakage of the vascular fluid containing serum protein which has the antibacterial activity, right? And it will influx the phagocytic cell to the affected area, so that the phagocytic cell will come to the affected area and it can phagocytose the incoming pathogens. That we will discuss it right now. So this is a very good image how your immune system works. These green things are bacteria, E. coli, right? This green rod-like shape, and this pink thing is dendritic cell, which is again a phagocytic cell, right? And this blue color is macrophage. So what happens when this phagocytic cells will be exposed to green color bacteria, then they will do the phagocytosis. They will engulf the cell inside them and it will kill, right? So now so we just discussed here that inflammatory barrier, when your tissue is damaged or there is any infection, for example you have a cut in your tissue, right? Or suppose you are exposed to some pathogens, this induce the leakage of the vascular fluid. So how does it happen? Let's see. So this is the place where your tissue is damaged or infected, right? So it is your affected area, right? So now through this area bacteria will come and it will enter into your body, right? And this is your blood stream. Okay, so in the blood stream, as I told you that there are some myeloid cells, for example dendritic cell, monocyte, then macrophage, sorry, not macrophage, dendritic cell, monocyte, right? This granulocyte, they resides into the blood fluid, right? Because they are coming from myeloid progenitor, so they will be from so they will reside in the blood fluid. So when there is a tissue damage, then there will be a leakage in the vascular fluid. Vascular fluid means this blood stream. So when there will be a leakage, then this blood will come, right? So this blood will come into the affected area. So tissue damage causes release of vasoactive and chemoactive factors which triggers a local increase in the blood flow and capillary permeability. So they will release some molecules which will induce this capillary permeability. So what is capillary permeability, anyone? Any guess? Capillary permeability?
The permeation of blood through the capillaries.
Yeah, so from blood vessel to tissue, right? That is your aim. Your aim is to recruit your immune cells to the tissue from the bloodstream. Am I right? So your aim is to recruit the immune cells from your blood to your tissue, right? So this is called, so this thing requires the leakage of the vascular fluid or the capillary permeability. So when it happens, then the blood from your blood vessel will eh, try to enter into an affected area. So these permeable capillaries will allow an influx of fluid and cells. Cells means immune cells, right? For example, dendritic cell, monocytes, right? So they will enter into the affected area. Then phagocytes migrate to the site of the inflammation. Phagocyte, for example, macrophage, okay, neutrophil, they are all phagocyte. They will come into the, they will migrate into the site of the inflammation here, and then they will phagocyte, and so here the phagocytic cell will do the phagocytosis, and they will engulf the foreign particles, and they will secrete some antibacterial materials or exudate which can destroy the bacteria, right? So that's how your so this is also called as the capillary leakage or extraction, when your immune cells from the blood system, they will enter into the affected area where they shows the antibacterial activity or anti-pathogen activity, or sometimes they do some phagocytosis to protect yourself from the infected pathogens, right? So now if you will focus on the phagocytosis, then how does it happen? This, suppose this is your phagocytic cell. This phagocytic cell, how it will engulf the pathogens, right? So the process is, first bacteria will become attached to membrane vagination called pseudopodia. First this membrane will take a shape like this by the process of membrane invagination. So it is called as pseudopodia. So first this bacteria will be attached, or it will be trapped in this pseudopodia. Now this bacteria will, or this pathogen will be entered into the cell by the process of phagocytosis, right, in a phagosome. So now the bacteria is ingested, and it has formed the phagosome. Then phagosome will fuse with the lysosome. What is lysosome, anyone? What does it contain?
Sir, bacterial cell wall.
Huh? Sorry? Lysosome, what does it contain?
Sir, it lyses the bacterial cell wall.
Okay. How?
Hydrolytic enzymes.
Yes, it has
protease enzyme.
yes, it has many proteases and hydrolase enzymes, right? So at the pH, how is the pH of the lysosome? Acidic, basic, acidic, very much acidic. So that's why in this acidic pH your acid hydrolases are active. So now they can lyse the pathogen, right? And it can ingest the cell wall. So that's why this phagosome is fused to the lysosome, right? So which is called as a phagolysosome, where the this pathogen can be digested. So lysosomal enzymes digest the captured material or pathogen, and this digested material products will be released from the cell, right? So that is the process of uh, phagocytosis. That's how the cells will engulf pathogen, and it will release the, and it will digest the pathogen inside this lysosome or phagolysosome, and then it will release into the external environment, right? So that is the process of phagocytosis. Now you tell me if you have any doubt or not. Any doubt? Anyone any doubt?
Sir,
No, sir.
sir,
Yeah, tell me.
sir, I have watched lecture videos, sir, and still I don't know how to collect the lecture notes, sir. Is there any way for collecting lecture notes as a text?
Lecture note. Uh, I mean, which lecture note? Can you specify? Uh, who is this? Hello? Who speaking? Hello? I'm not able to hear your voice. Okay, so lecture note actually, I mean, you are asking for which lecture note? The one which is already submitted in the portal. Okay, I think her mic is not working. I am not able to hear. Okay, so continue. Uh, any doubt from the slide? Okay, how to collect the lecture notes? Uh, which lecture note? Okay, how to collect the lecture note? I think the transcript is already there in the portal, right? You can follow the transcript for that. But the material, I don't think it will be available. I'm not sure, but I don't think it's there. But transcript is there. I think you can check for the transcript if you need that. Okay, so let's back to the our topic. So this is the way uh, by which your when there is infection, then your system, then your cells in your immune system will go and it will give you the protection against the infection, and that's how uh, the phagocytic cell will come and do the phagocytosis, right? So now we will shift to adaptive immunity, right? So now if I will ask you that uh, how does the adaptive immunity cells, for example B cell, TH cell, TC cell, how do they train? Any idea? Or who trained them? Or what is the process of training? Training. Can you please respond? Am I audible or my voice is not clear? Can anyone please confirm? Bhanu, I can't hear you.
Sir, your voice is clear. Go ahead, sir.
Okay. Okay. So the question is, uh, huh. So the question is that the different cells for adaptability, B cell, TH cell, and TC cell. So how do you think that what is the process of training them? That's why they are adaptive. How do they adapt? Anyone want to go with that?
The antigen.
Through the antigen. Okay.
Antibody interaction of antigen antibody. Sir.
So how do they know that which antibody is, suppose for example for B cell it secretes antibody, right? So but how do they know that which antibody is suitable for this particular pathogen?
That's why it helps for T cells.
Memory cells have this specific structure for suitable antibodies.
But how do this memory cells formed? How do they know that this is the perfect antibody for this antigen?
Because they have memory cells.
Memory cells is fine, but when I'm talking about the production of the memory cell, so this memory cell has some information, right, that which antibody is required for this kind of infection, right? So how do they know that which antibody is perfect? How does this memory cell form? Before that it needs to decide that which antibody should be there. So that's I'm asking. The process is that we will discuss that uh, B cell, they can also present antigen through this B cell through their uh, MHC class 2 protein, right? So these antigens which are presented by the MHC class 2, they can use to fit with the sorry, actually B cell, they will first of all they will produce a range of antibodies, antigen binding receptor or antibody, which are surface bound, right? So this surface bound antibody will test the interaction with the particular antigen by some antigen presenting cells, for example follicular dendritic cell. We will go to that. The follicular dendritic cell will present the antigen, right? And this antigen will now so after that the antigen will be bound with this B cells expressing the antibodies, right? So now this B cell has various variety of antibodies. So now now the antibody which will be able to perfectly bind with the antigen which is presented by the dendritic cell, then this particular antibody will be treated as that, yes, this is the correct antibody. So only those B cells which can express this particular antibody, they will be survived, other will be die, right? So that is called clonal selection. B cell will make a multiple number of clones, right, with diversity of antibody. But which B cells will survive? The B cells uh, which will express the antibody which can make a interaction with the antigen presented by the antigen presenting cells or APC. So only those B cells can be selected and they will be differentiated into two cells. One is plasma B cell which will secrete antibody, and another one is memory B cell, right? So that's how this memory B cell form, by training it. How and what is the training procedure? It will fit the antibody with the particular antigen, and then it will check that, or it will screen the antibodies which is actually fitting with the antigen. Only those B cells will survive, others will die, and this survived B cell who were selected in this process called clonal selection, they will differentiate into memory B cell and plasma B cell, right? So that's how they be trained. So here, if you will see that there are two response in this adaptive immunity. One is first time response, primary response, and another is secondary response. So why the second? So what is the secondary response? Any idea? Why this secondary response? Or what is this? What is this secondary response?
Sir, it happens due to the repetitive attack of the same microorganisms.
Yes.
And it occurs with our body comes with an army of antibodies against those microorganisms, sir.
Yes, because there is already memory B cell, right? So that's why the cell will not take time to train, because training is already done. Now in the second time infection or secondary infection, now the cell doesn't need any training. It already has the memory B cell, so it will just start secreting the antibody. That's why the antibody level is more in the secondary immune response, right? And here, if you will see that there are two kind of response in the adaptive immunity. One is cell mediated response, one is humoral response. So what is cell mediated response? The internalized antigen, they are digested by the cell wall, which we just discussed in the lysosome. The cell wall was digested, as we discussed in the last slide, right? So that's how the antigen will be digested. It will be degraded into several particles, and each particle will serve as an antigen, and this altered self cell present those antigen by either class 1 MHC or class 2 MHC, right? T helper cell is an example of a cell which present the, or B cell, they can present the antigenic this antigen by class 2 MHC, and TC cell is the example who present the antigen by class 1 MHC, right? And this binding and when they will interact with the B cell, they will activate the B cell, and the activated B cell will start differentiating into the, so as I told you that they will first start expressing the several different type of antibody, and they will fit the antibody with the antigen which is presented by follicular dendritic cell in the lymph node, and the antibodies which are fitted with the antigen, those B cells will survive, others will die, right? So this is the process of adaptive immunity, right? So what is humoral response? When B cell, it will expose with the antigen, so this B cell will interact with antigen and differentiate it into the antibody secreted plasma cell, right? As I explained you, and I also after this I will discuss it, the entire process, how this B cell is trained and it will differentiate it into the plasma B cell and memory B cell. So when it interacts with the antigen, it will be differentiated into the antibody secreting plasma cell, and it will secrete antibody. Antibody will bind the antigen and facilitate its clearance from the body. That is humoral response, right? And this response, which is mediated by the, for example, here cytotoxic T cell. This cytotoxic T cell, when they for example, when they these T cells and the T cell receptor, they recognize the antigen bound to MHC molecule, right? And binding antigen MHC, this interaction activates the T cell, and activated T cells secretes the cytokines that contribute to the activation of B cell, TC cell, and other cell. So what's happening here again? T cell receptor recognize the antigen bound to the MHC molecule. Binding antigen MHC activates the T cell. This activated T helper cell secretes cytokines which will again activate the B cell, TC cell, and other cell, right? This activated TC cell or cytotoxic T cell, they will recognize and they will kill the cells which were infected with antigen, right? Those cells will be killed by cytotoxic T cell. So it will basically secrete the granzymes and perforin, which will help to destroy the cell which is infected. So this is cell mediated response, and this activated TH cell will also activate the B cell. This B cell will secrete the antibody, right? This antibody will bind to the antigen and it will clear the antigens from the body. So this is antibody mediated. This clearance of antigen is antibody mediated. That's why it is humoral response. And this clearance of pathogen, this is cell mediated or CTL or cytotoxic T cell mediated. So that's why it is cell mediated response. So again, so now if I will ask you, what is the difference between cell mediated response and humoral response? Anyone? Cell mediated response and humoral response. Difference? Anyone want to try? Did you understand?
Humoral response in body fluid, cell mediated in some type of cells which are
Cells?
not
Yes.
flowing in the blood or tissue fluid.
No. Here see T cells. No, the thing is not about blood or tissue or lymph. The thing is about how they are killing. So can you point out any difference in the process of killing? Cell mediated and humoral response.
So in cell-mediated immune response, it involves the phagocytic nature of the cytotoxic T cell. In humoral
Wait,
response, it involves
wait.
the production of.
Just a minute. Cytotoxic T cell, it does not have the phagocytosis property. Okay. So please don't be confused. Okay, don't do this mistake. Cytotoxic T cell, they don't phagocyte. They are not phagocytic cell. They secrete some molecules, for example, perforin and granzyme. That toxic molecule will kill the infected cell. Okay, so it will not phagocytosis. Okay, then continue.
Sorry for misunderstanding, sir. I
No
got
problem.
it, sir.
Huh.
Then humoral response, it involves the antibody production.
Yes.
This antibody attaches to the antigen, and
Yes.
then antibody interactions happens and leads to the destruction of the antigen, sir.
Very good. Yes, so this is humoral response, and for cell mediated, when this cytotoxic T cell, for example, when these CTLs are activated, they will recognize and they kill the infected cell. So this killing is mediated by the cell, which is cytotoxic T cell, and this killing is mediated by the antibody. That's why it is humoral response. Okay, so that is the main difference between cell mediated response, which is mediated by cytotoxic T cell or other cell, for example NK cell. All of these are cell mediated response, but this response which is mediated by the antibody, this is humoral response. Here B cell will be secreting the antibody, this antibody will clear the pathogen, right? So this is the basic difference between the cell mediated response and humoral response. This is very important to know how to differentiate between these two responses. Okay, so now if I will ask you a question that for example, the immune cell that bridge between innate and adaptive immune response. Can you tell me?
Sir, helper T cells.
Helper T cell. No, helper T cell is not for
Dendritic cells?
dendritic cells. No, dendritic cell is for only innate immune response. No? So if I will give you four options: macrophage, B cell, dendritic cell, T cell. Macrophage, B cell, dendritic cell, T cell. Which one? Someone said na dendritic cell. Who was this?
Yes, sir.
Okay, okay, you. So,
Yes, sir.
do you think that the dendritic cell is the right answer, or you need to modify it? Some. The actual answer is follicular dendritic cell. So what happens? That also we will discuss in the last slide. So what happens? The follicular dendritic cell or FDC, they go to the, suppose if I will try to draw something. Wait a minute. So this follicular, this is your FDC. This is follicular dendritic cell, right? This has some projection. So when there is a infection site, it will go there in this infection site, right? This is your infection site. So it will go there and it will internalize the pathogen. Suppose there are some pathogen, so it will phagocytosis the pathogen inside this, and it will present those pathogen because it is a APC, antigen presenting cell. So these pathogens will be engulfed and they will be presented in its body, right? So that's why it is follicular dendritic cell. So it has the antigen presenting property. Okay. So now when they will present the antigens which actually infected the body, so after this they will go to the lymph node, right? In lymph node, where are they? Lymph node contains B cell, T cell, right? So now in the lymph node it will come and it will present this antigen. So now this B cell will come. This is your B cell. B cell, that I have already told that it will express the antibody in the surface, right? So now this surface exposed antibody and this antigen presented by the follicular dendritic cell, they will make an interaction. If this interaction survived, then this B cell will survive. If this interaction weak, then this B cell will die, right? This is called clonal selection, which is a part of the adaptive immune response, right? This is selection. The B cells, it is expressing antibody. This antibody, if it can fit with the antigen presented by the APC, which is here follicular dendritic cell, if it can fit with this antigen, then these B cells will be selected and they will differentiate into the memory B cell and plasma B cell. But if not, then it will be die, right? So in this way, follicular dendritic cell or FDC, they can play role in the adaptive immune response, right? And okay, apart from that, you also know its role in the innate immune response, because it is a phagocytic cell. It can, that's why it can take part in the innate immune response. It can phagocytosis the pathogens and they can kill the pathogens. So that is its contribution in innate immune system, and also it can contribute into the adaptive immune system, right? So now I want some of you to explain me why the dendritic cell is the common bridge between them. So anyone want to come? Shivani Tomar has written something. Acha, difference between the cell mediated. Yeah, you are absolutely right, Shivani. So now the this question, can any of you explain to me why dendritic cell or follicular dendritic cell, it plays a role in bridging the innate and adaptive immune system? Whatever you understood. Lohita, can you explain? Are you there, Lohita? Poonam Raut?
Sir, may I tell?
Yes, tell me the difference. I mean the why it is a bridge between between adaptive and innate.
Sir, in innate immunity,
Yes.
phagocytoses the pathogens.
Yes.
At the same time, it acts as an antigen presenting cell.
Very good.
And it helps for the clonal selection, at the same time for the antigen production from the B cells, sir. So through that,
Antibody
adaptive
production.
immunity is happening.
Antibody production from B cell, not antigen.
Okay, sir.
Yeah. Yes. Correct. Perfect. Okay. So okay. So the next question is, hematopoietic stem cells are pluripotent, which means that they are antigen specific cell capable of developing into any blood cells, not self renewing, committed to produce cells of a single lineage. The statement is hematopoietic stem cell, which we showed here. The hematopoietic stem cells, they are uh, pluripotent. So what does it mean? What do you mean by pluripotency? Anyone? Pluripotency? Who will tell me the pluripotency? Hari Shankar? Hari Shankar, you there?
Pluripotency, it's a multiplication of
Multiplication of what?
multiplication of cell immunity.
No, pluripotency is when a cell can be differentiated into any type of cell, right? That is pluripotency. It can be blood cell, it can be other tissue, anything, right? Yeah, SA what is right, they can be able to become any type of cell. Yes, so if hematopoietic stem cells are pluripotent, then option A, they are antigen specific cell. Is it right or wrong, this statement? They are antigen specific cell. Is it right or wrong?
Yes.
True or false?
It's true.
No, it cannot be true, no? Because pluripotent means it can be differentiated into any type of cell, right? But option A is saying it is antigen specific cell, but that is not true. It can be differentiated into any type of cell. Yes, Sudha B is saying wrong. Yes, it is wrong. How do you understand why is it wrong? Because pluripotency means you can differentiate into any type of cell. That's why. Okay. Option B, capable of developing into any blood cell. Is it true or false? Capable of becoming capable of developing into any blood cell. True or false? True. Sudha is saying true. It is true because it is a pluripotent cell. Okay. 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 So the next question is, acquired immunity shows all of the following except uh, non-specific to particular pathogen. Is it right? Regarding acquired immunity or adaptive immunity, so first option is non-specific to particular antigen. Is it right?
False.
False. Why is it false?
It is specific.
Yes, it is specific. That's why it is adaptive, right? So yeah, option B is diversity. Is it true for acquired immunity? Selva is saying false. Yes, it is false because it is specific. Right? Option C, self or non-self recognition. Is it true?
True.
No. Why no? False. Yashomita say it is false. Okay, so Selva and Yashvita. So, Selva, why is it false? Why is it false? Yashvita, why is it false? Rajesh V, are you there? It may take some time, maybe wrongly for a self. No, essay what yeah, so you are actually, the thing is, uh, see, acquired immunity will distinguish between specific antigen. Suppose you are infected with two different antigen, right? But your acquired immunity that knows only about the antigen B. Suppose you are infected with antigen A and antigen B, but you have only adaptive immunity of antigen B, right? So it will only produce response when it will be exposed to antigen B, not antigen A, right? So that's why option A is right. That I mean option A is false. That it is not specific to particular pathogen, right? It is specific. Option B is diversity. So again it is false because yeah. So the third option is self and non-self. Yeah, self and non-self recognition and memory. So why this self and non-self recognition? So it is true actually. Why? Because it can recognize that which is self antigen, which is non-self antigen. Which is self and which is non-self antigen?
Non-self antigens are the any foreign particles.
Yeah, which is not from your body, right?
Yeah.
That is so yeah. So
So
this
sir,
is,
it is true now.
yeah, it is true. Acha, sorry for the mistake I did. I think my mistake said it false, but it is true. Self or non-self recognition. Yes, it is true because it can recognize between the self and non-self. Option D, memory. Is it true, memory? Memory. Shivani?
True.
True.
Memories.
Yes, because it can produce memory B cells, right? So it can keep a memory of the previous or last, you know, infected cells, right? So yeah. Next question is, the common myeloid progenitor cells give rise to common myeloid progenitor cell that give rise to option A, neutrophil alone. Is it true or false? SA,
False.
yeah, SA what said false, Shivani is saying false, Sudha is also saying false. Okay, it is false because not neutrophil alone, it is Shubhas is also is right. So not, it is only neutrophil, along with that many things are there. So neutrophil alone is false. Neutrophil, basophil and NK cells. True or false?
So can you repeat?
Neutrophil, basophil, natural killer cell. Sudha is saying false. Okay. Others? Subhash is false. Okay.
False.
Essay what? Why it is true? Arya is also saying false. SA what? Can you switch on your mic? Or if your mic is not working, you can also explain in the chat. Why is it true? If we go back to the slide, see, myeloid progenitor, is it giving NK cell? No, right? NK cell is given by lymphoid
Lymphoid.
progenitor. Very good. So this is not by myeloid progenitor. So that's why it is false. Okay. So option C, B cell, T cell and NK cell. True or false?
False.
False. Of course false, because all of them are by lymphoid
Lymphoid.
progenitor. Option D, neutrophil, basophil and monocyte. True. Yes. Shubhasis is saying true. Others? Okay, Sudha is saying true. Shivani?
So can you repeat?
Neutrophil, basophil, monocyte. Arya is saying true. Essevat is saying false. Okay.
So why is it true?
No. What do you think? That's what I'm asking. Poonam is also saying true. Neutrophil, basophil, monocyte. Shivani, you understood the question?
No. No, sir.
Same question. The common myeloid progenitor cell give rise to, the option is neutrophil, basophil, monocyte. Is it true or false?
Okay.
Is it true or false?
So I got it now. True.
True. Okay.
True.
Say what? You said false. Why? Selva is saying true. Selva, why is it true? Because it is from this previous chart we read, there we found that all granulocyte, neutrophil, basophil, monocyte, all of them are granulocyte. They are their lineage is myeloid progenitor cell. So option D is right. Okay. Okay. So the next question is the example of secondary lymphoid organ. Okay. First option is spleen and thymus. Option A. Is it true or false? True. Why is it true? False. Why true,
Sir,
why
also
false?
repeat the question, sir.
Example of secondary lymphoid organ. Option A is spleen and thymus. Are they?
True, sir.
It is not true.
Sir, false.
Yes, Sudha is right. Thymus is the primary lymphoid organ. Spleen is the secondary, but
Spleen is secondary.
secondary and thymus is primary lymphoid organ, right? So it is false. Option B, lymph node and thymus.
False.
False, because thymus is there. Spleen, yes. Spleen and GALT.
True.
True, yes. GALT, yeah. GALT is secondary lymphoid organ. Okay. So what is the full form of this GALT?
Gut associated
Gut
lymphoid
associated.
tissue.
Yes, it is the I think one of the biggest lymphoid tissue system.
Yes, sir.
Option D. So it is true. Spleen and GALT. Option D. Bone marrow and malt.
False.
Why false?
So bone marrow is
Primary, right? Yes, very good. Yes. Malt is secondary, but and again, what is the full form of malt? M for
mucus
mucus.
associated lymphoid.
Very good, very good. So bone marrow is primary, so again it is false. So option C is right, spleen and GALT. Right? Okay. So now we again go back to our topic which we are discussing. So we discussed till and here also you can see the electron micrograph of rod shaped E. coli, which is adhered to the surface of epithelial cell of urinary tract, and it induces the innate immunity response. So we are here, adaptive immunity, right? So now let's talk about macrophage. So macrophage is a phagocytic cell. When monocyte enter into the tissue, then this monocyte become macrophage, right? Remember we discussed?
Yes, sir.
Yeah. Okay. There is some connectivity problem, I think. Yeah. So that is macrophage, where monocyte gets into the tissue, right? So now, and macrophage is a phagocytic cell. So the process into the phagocytosis is, again, it is forming some pseudopodia. But this bacteria will come and it will adhere to the pseudopodia, which is a projection of the macrophage cell membrane. So bacteria will come and it will trapped here in this pseudopodia. After that, it will be ingested to the macrophage by phagocytosis. Then and it will be inside the phagosome. This phagosome will be fused to lysosome to make the phagolysosome, and inside the phagolysosome there are two possibilities. One is this enzymes in the phagolysosome will degrade the this antigen, and this antigenic fragments will be released, which is called the exocytose degraded material, right? Another possibility is as macrophage has an antigen presenting capability, it is a APC, so here this class 2 MHC will bind to the antigenic fragment, one of the antigenic fragment, and it will present the antigenic fragment. So it can do two things. It can degrade the antigenic fragment, and it can exocytose this degraded material. Alternatively, it can also present the antigen so that the B cells, T cells, they can be activated, right? So it has two properties. And the killing of macrophage is either oxygen dependent or oxygen independent. So oxygen dependent killing involves where the macrophage has oxidase, right? This oxidase will reduce the reduce the O2 to toxic this anions of oxygen. O2 minus OH, which is hydroxyl radical H2O2 hypochlorite ion, all of them are toxics and they can make the oxidative stress into the antigen. Right? That's how they kill. Or it can also form reactive nitrogen intermediate, nitric oxide, nitrogen dioxide, nitrous, and NH2Cl, so that it will make a toxic effect into the internalized antigen, and that's how they can kill the antigen. Another thing is oxygen independent killing. So it can be mediated by some chemicals or some peptide which is defensive, TNF alpha, lysozyme, which is example of here in the lysosome, and hydrolytic enzyme. They can digest the cell wall and it can digest the antigen to the antigenic fragments, right? So this is an example of oxygen independent killing, and there is also occurrence of oxygen dependent killing where oxygen is reduced to this free radical or superoxide anion, which can actually put the oxidative stress. So the internalized antigen will be killed, right? So that is the macrophage, its way of killing. And also this macrophage has a TLR or toll-like receptor. This toll-like receptor are the pattern recognition molecule. They can recognize the antigen and they can actually place a role of receptor. So this TLR will recognize the antigen and then this antigen will be phagocytosed by the macrophage. And after that this kind of killing will be there. Oxygen dependent and oxygen independent. Right? Any doubt?
No, sir.
Okay. So now granulocyte. So this eosinophil, neutrophil, all of them are from myeloid progenitor. So they give rise to the granulocyte. Why they are called granulocyte? Neutrophil, eosinophil, and basophil.
They are granules having the enzymes.
Yes. When they recognize the antigen, then they will release these granules from this here, right? So these granules will help to neutralize the antigen. Okay. Again here we already showed that hematopoietic stem cell will differentiate into the myeloid and lymphoid. This myeloid will give rise of intestinal dendritic cell, Langerhans cell, and this monocyte, also monocyte. This monocyte will give rise of myeloid dendritic cell and lymphoid dendritic cell. So these are the different kind of dendritic cell, right? So as we already showed there, that dendritic cell is a cell which is generated from as well as myeloid as well as the lymphoid progenitor, right? From the myeloid, it first monocyte, then this monocyte can be differentiated into the myeloid dendritic cell. Also it can give rise to the intestinal dendritic cell. Also it can give rise to Langerhans cell, right? And this lymphoid progenitor can give rise to the lymphoid dendritic cell. So if I will ask you, name a cell, immune cell, which is from both of the, which is common for myeloid progenitor and the lymphoid progenitor, then what will your answer be?
Dendritic cells.
Yes, dendritic cells. Okay. So now let's talk about antibody. So antibody, I think you are familiar with this antibody Y-shaped structure, right? So how many chains antibody do you have, and what are they?
Light chain, heavy chain.
Light chain, heavy chain. How many light chain? How many heavy chain?
2 light chains.
Two light chain and a heavy chain?
4 heavy chains.
No.
2 heavy chains.
Two heavy chains. These two are two heavy chains and these two are two light chains. Okay, so this light and heavy chains are connected by this disulfide bond here also, and these two heavy chains are connected by this two disulfide bond, right? It is called the hinge region. So if you want to break the disulfide bond, then what will happen? This heavier light chain will be separated, these two heavy chain will be separated, and this heavier light chain will be separated, right? So which reagent you use to cleave the disulfide bond? Any idea? If you want to cleave the disulfide bond, so which reagent you will use, and how many fragment you should get?
So 4 fragments.
4 fragments. And which reagent you will use for that?
Sir, maybe I'm not wrong. I'm wrong. Like proteolytic enzymes like pepsin.
No, not pepsin. For disulfide bond, which reagent? It is very important. You should be aware about.
Mercaptan.
Yes, mercaptan. It will reduce this disulfide linkage, right? So that's why this bond will be broken, and you will get 4 fragments like this here, right? Now, if you will digest the antibody with pepsin, then how many fragments are? What are they? Pepsin digestion, how many fragments and what are they? One fragment. Yes, so one intact fragment, and actually you will get multiple number of fragments, because if you will digest with pepsin will just cut here, okay, here. So if it will cut here, you will get this fragment. Also, this pepsin in this FC region, pepsin will cut non-specifically. Okay, that's why this FC region will be completely fragmented, right? So if you will, so you will get lots of FC fragments, and one this FAB prime 2, right? Why this is FAB prime 2? Why is not FAB? Why is it FAB prime 2? I think you know about the FAB and FC, right? Do you? Antibody FAB region and FC region, you know?
Yes, sir.
Yeah. So this region is FAB, right? And this is FC. AB means antibody binding. This region is responsible for binding of antibody antigen, right? Right. So that's why it is FAB, and this is FC, right? So why is it FAB prime? Why is it FAB prime? Why not FAB? Anyone? Anyone? FAB prime because its cutting site is here. So it is taking entire FAB. This much is FAB, right? This is this one FAB. This is another FAB, but it is cutting here, right here. So it is taking FAB plus the hinge region. This black one is the hinge region, right? So it is taking FAB plus the hinge region. That's why it is FAB plus hinge. So it is FAB prime, not FAB, plus FC fragments. Okay. So what will happen if you will digest with papain? Papain digestion.
3 fragments generated.
How many? 3.
Yes, sir.
Okay. So what are they? It will cut here, right? It will cut exactly here. So how many FAB fragment and how many FC fragment?
2 FAB fragments and 1 FC fragment.
So if you will, okay, yes. So if you will run the gel, then how many band you will see? For this case, papain digestion. No, for this case, papain digestion. Okay, so it will be your assignment that you just think it, or you can study also, that each digestion, pepsin digestion, peptide digestion, and markapritch ethanol digestion, how many bands you will get when you will run it to the gel. Okay, so we can discuss it in the next class. Okay, so let's come back to some question again. 5 more questions are remaining. Okay, so the first question is strategies applied by host to deal with the microbes threat. Option A, avoidance. Option B, resistant. Option C, tolerance. Option D, all of the above. Which one is correct?
All of them.
Yes, avoidance, resistance, and tolerance. What do you mean by avoidance? Yes, option D, Arya. What do you mean by avoidance? Avoidance means you are ignoring, right? So it is not affecting you. Option B is resistance. The cell when the immune system will actually will try to resist, to prevent, right? Option C is tolerance. Both all of these 3 are required. So option D is the right answer. So next question is an inflammatory response is induced by sensor cells through the production of mediators called, option A, cytokines only. Is it correct? Inflammatory response is induced by sensor cells through the production of mediators called, option A, cytokines only. Is it true?
So can you please repeat?
Inflammatory response is induced by the production of the mediators. Which mediators? Option A, cytokines only. Is it true?
False.
False. Option B, chemokines only. Someone else? Chemokines only.
So false.
False. Okay. Option C, cytokines and chemokines both.
True.
True. Yes. Always inflammatory response will be mediated by the cytokines and chemokines, not only cytokines. Okay. So the next question is, the term vaccination was coined by option A, Louis Pasteur. Is it true?
False.
False. Edward Jenner.
Yeah, true.
True. Okay. Okay, so the option B is correct, right? Next question. Okay, that we discussed, right? Acquired immunity. Individual exposed to smallpox virus and immune to same disease due to option A. So when someone is exposed to smallpox virus, but then when after that they will be immune to the same disease. The reason is option A, presence of larger quantities of antibodies. Is it true? Presence of larger quantity of antibody. Arya is saying true. Others? Pushpa? Paulavi? Shivani? Shivani Tomar?
Sir, I don't know.
Okay. Rajesh? Sudha?
Sir, can you repeat again, sir?
Individuals exposed to smallpox virus are immune to the same disease. Why? The reason is option A, presence of large amount of antibodies.
Sir, it might be due to secondary immune response, sir.
So,
So,
huh?
in secondary immune response, we get a large amount of antibodies, sir.
Yeah.
Due to
But
presence of memory cells.
yeah, so which one will be correct? Due to large quantity of antibodies or for memory cell? Large quantity of antibody comes from the memory cells, right? So the option is
Memory cells.
yes. Option is the answer is memory cell. Option A was presence of larger quantity of antibody. Option B was presence of long-lived memory cell. The answer is the long-lived memory cell. Why? Because of the presence of the memory cell, the cell was able to combat with the secondary infection, right? So that's why it is true. Okay, so I think the questions are done. So we are left with few portion of our topic. So let's complete it. Okay, so antibody we discussed. Then we will go to so there are different types of antibody: IgG, IgD, IgE, IgM, which exists in a pentameric format, and IgA. IgA is an antibody which is also called as the secretory antibody. It can secrete, right? So that's why when it can secrete it through your eyes, through your tears, through your uh, saliva, so that's why it is secretory antibody, right? uh, IgE importance is what? IgE when it has a role in the allergic response, right? So it can bind to the basophil mast cell, right? Because mast cell has a antibody receptor. So when this IgE antibody is bound to some allergen, then this antigen antibody complex will bind to the antibody receptor of mast cell, and this will induce the release of the granules of mast cell, which are basically histamine and basophil also. So it happens with basophil and mast cell. So they will bind, IgE will bind to them, and they will release histamine. That's why the anti-allergic drug you find, they have the antihistamine inhibitor to inhibit the allergic response. And as I told you that IgA is the antibody which can secrete, so it can secrete. So this is the way. So here plasma cells secrete the IgA antibody, which is dimeric here. So this dimeric antibody will be secreted by the cell, and then it can the property to cross this epithelial cell, and it can go to and it can be secreted. right? So this is antibody which can be secreted, and yeah, and this is the antibody IgM which exists in a pentameric form. So there are three terms: isotypic, allotypic, idiotypic. Anyone knows the differences? Isotypic means, suppose there are two antibodies with different classes. One is of IgG1, one is from IgM, right? So that's why they are different. So they are called isotypic isotypic determinant. Why? Their Fc region is different. They have different isotype. One is IgG1, one is IgM. Next is allotypic determinant. So there are two antibodies. Their isotype is same. That is IgG1, but they are from two different strains. One is from strain A, one is from strain B. That's why they are different, right? See, if you will see this IgG1, so this region is different from IgG1 to this strain A to strain B, right? See, there is a difference between this strain A and strain B, right? So these two antibody are of same class, so their isotype is same, that is IgG1, but their allotype is different because they are from two different strains, and that's why they are different. So they are allotypic variant. Third is idiotypic variant, means two antibodies which are from same isotype IgG1 and which are from same allotype, right? But they differ in the variable region. Variable region, which is also known as the paratome, it binds to the epitope of the antigen, right? So that region is responsible for binding to the antigen, right? So that's why it is a variable region. And when the two antibodies, they are of two same classes or same isotype and two same allotype in the same strain. Suppose example, for strain A we are getting IgG1, so these two IgG1 are from strain A, right? So their allotype is also same, but this antibody is against antigen A and this antibody is against antigen B. So that's why their variable region is different, right? So that's why they are called idiotypic determinants. Any doubt? Any doubt? Okay, now we will see the antigen antibody interaction. So antigen region which is interacting, that is known as epitope, and antibody region which is interacting, that is known as paratome, right? So this antibody antigen interaction, how does it happen? By this hydrogen bond, ionic bond, hydrophobic interaction, van der Waals interaction, and ionic bond, right? And there is antigen antibody reaction to, there are three zones. One is antibody excess zone, one is equivalence zone, one is antigen excess zone. So antibody will be precipitate when, if you will plot a graph with antibody precipitate and antigen added. So suppose initially there was no antigen, so now we are gradually starting adding the antigen. So now initially there is excess antibody plus, but there is no antigen. So gradually when we will start adding antigen, this interaction will start happening. Then it will reach to the equivalence zone when the antibody and antigen both of will be same amount, right? Then they will make a cross link, and that will help them to precipitate. Then you will get a precipitation here, right? Again, if you will keep adding the antigen, then what will happen? Then it will come to the antigen excess zone. So here antigen now is more, so now again it cannot form the cross link. So antigen precipitation will again decrease. Okay, so understood? Antibody excess zone, equivalence zone, and antigen excess zone. Okay, so yeah, so that I discussed with you, that what is clonal selection. So again, if you will see that this is the process of activating a B cell. So when B cell will start presenting the antigen by this class 2 MHC, then this will be recognized by a helper T cell by TCR or T cell receptor. So when this interaction established TCR and the MHC class 2, then there are some co-stimulatory signal. For example, B7 from B1 interact with the CD28 from T cell, and CD40 of B cell will interact with the CD40L from TH cell. Now this is the complete way of activating the B cell. Now this B cell will be activated and it will secrete the cytokine, and this cytokine secretion will activate the B cell, because now B cell will start proliferating. So that is the way of activating the B cell. So now so now after getting lots of B cell, how will you select that which B cells will survive? That is clonal selection. How does it happen? So in this lymph node there are 3 zones: dark zone, dark zone and 2 light zone. So here when the activated B cell will come, they will start presenting the antigen. So after activation they will start expressing the antibody here after activation by helper T cell. So when T cell will activate them, then they will proliferate. They will make lots of clone of them, and they will express the antibody in the surface. So now this antibody will be fitted into the antigen which is presented by this follicular dendritic cell. Okay, that happens in the light zone, right? So now this antigen antibody complex interaction will be checked here, and the B cells which are having the proper antibody which can make a strong interaction with the antigen, they will survive. They are selected, and rest will die. So here you can see there are 2 different kind of B cell. One is one is expressing the blue antibody, another one is expressing the black antibody, right? So the B cells with the blue antibody, they are making stronger antigen antibody interaction, so they will select it. And the B cells which has the black antibody, they are not making the proper interaction with antigen, so they will not be selected, so they will die. So these B cells will be selected by this clonal selection method, and this B cell will be activated by the helper T cell here, and this activation process is described here. How does it happen? So now this activated B cell will start, this B cell which is selected, it will now start proliferating, right? And it will be differentiated into memory B cell and plasma B cell. Memory B cell will be kept inside the cell, but this plasma B cell will start secreting the antibody, right? So now this antibody will bind with the antigen, and it will show its effect. What is humoral response? What is cell-mediated response? So this antibody will basically show the humoral response, right? So that is the process of clonal selection, where the particular B cells which has antibody which can fit properly with antigen presented by the dendritic cells here, those antibody containing B cell will be selected, and they will be activated, and they will be secrete the antibody, right? That is the clonal selection we talked about. Okay, so any doubt till now in today's session? Any doubt, please tell me. So any doubt? Are you going to ask any question? Hello? Anyone? Any question? Okay, Lohita is saying no. Okay, so I think it will be better that if you will uh, become more interactive. So is there any issue for mic? Is it not working?
So actually we didn't get you, sir.
Sorry.
Actually didn't get you, sir.
Yeah. So I was asking that that we can actually increase the interaction by speaking, because when I present the slide, then it is sometimes the chat are not visible, so I could not even see that who is asking what, right? So that's why it will be better if you can actually ask me with a mic. Is the mic all right with all of you or mine?
Yeah, sir.
Yes, sir.
Okay, okay, okay. Then yeah. So any doubt you have or from the
No,
test session?
No, sir.
Okay. So we will meet on next Saturday. So you please work with that question I gave you, that how many bands you will get, right, in pepsin digestion, papain digestion, and beta and mercaptrinol digestion. How many bands you will get when you will run a HDS page? Okay, that and then each of you will tell your answer. Then we can actually see that. Okay. So any question you have? Or any, I mean, any suggestion to me, if like anything I can add, tell me if there is something. Okay, so I think it's fine. So see you guys. We can meet on next Saturday. Bye.
Thank you.
Thank you for joining. Bye.